This is the poster titled Functional Profiling of Respiratory Tract Microbiomes Indicates Altered Microbial Pathways in COVID-19 Patients. My name is Niina Haiminen and I'm from IBM Research. The microbiome is a collection of microorganisms in an environment such as the human gut or skin, on food, on building surfaces, etc. So each microbe has the unique genome and genes, and the microbiome is also called the second genome, as it comprises millions of microbial genes and as many as over half the cells in our body are not human. So the microbiome interacts with the host, it changes in response to diet and environment, it impacts metabolism and even modulates response to medications. Alterations in the microbiome have been detected in many diseases, such as respiratory disease. High-throughput sequencing is used to investigate the microbiome, such as targeted single-gene sequencing or shotgun sequencing. Here we're focusing on metatranscriptomics, the RNA of the microbes. What is the community doing? The arrival of the previously unseen human pathogen SARS-CoV-2 also causes a shift in the host human microbiome. We would like to understand the science behind the response to inform disease management, and findings can also inform the design of probiotics or drugs to promote beneficial and reduce harmful microbes or microbial functions. In this analysis, we used a tool called PRROMenade, which we previously published for microbiome functional classification. So it takes as input the sequencing reads, in this case millions of reads per sample, using a reference database. Here the IBM functional genomics platform, from which we extracted 21 million bacterial and viral protein domains that have been annotated with KEGG enzyme codes, reflecting the activity of the microbiome. Using this PRROMenade annotation, we can get functional profiles, profile for each sample. We can compare the profiles across healthy and afflicted cohorts, and we can also build a mapping of functions to metabolic pathways and see the pathway differences. In this analysis that was published in Scientific Reports, we utilized data from Shen et al. For 8 COVID-19 patients, 20 healthy controls, and 25 community-acquired pneumonia cases. This data was total RNA sequencing of respiratory tract samples. So after obtaining the sequence data filtering, we applied PRROMenade functional profiling. And here, visually, we see in the multidimensional scaling how the different cohorts separate. So in this original analysis by Shen et al., they indicated there was something changing in the microbiome in COVID-19, but no specific microbes were detected. Instead of the microbial naming and taxonomic analysis, here we approach this from a functional analysis, so looking at the potential functions, and indeed found some differences between the cohorts. We investigated the differentially abundant features by rodeo by using the PRROMenade functional profiles as input. And we do see that the samples cluster by cohort using the top differential features, as indicated by the sample colors on the top row. Also, the features do cluster, and in the bottommost cluster you see lighter colors on the left, which indicates higher values in COVID-19. For example, for this one enzyme code that was also detected as increased in a metaproteome study. And this example was also highlighted in the recent Empress tool publication. Finally, we mapped the functional profiles functions onto pathways, and then we could compute a pathway score and indicate those pathways that are more or less abundant in COVID-19 compared to the healthy pneumonia cases. So the more abundant pathways in COVID-19 were found to often be related to carbohydrate metabolism, as well as the less abundant ones to lipid and glycan metabolism. Similar findings have also been indicated in recent literature in COVID-19 gut microbiomes. And these findings could potentially inform disease management, but further investigation of these pathways and microbes is certainly needed. I would like to thank you for your attention and leave you with these references with more information on the presented study and the methods and data used.